Originally from Lund, but I've been living in Gothenburg now for many years. And I'm working at this company called IRLAB, doing machine learning and a little bit of data analysis and a little bit of all kinds of things. Have you started recording? Yeah. I don't see any pop-up. But
no, I don't know.
Okay, so what's IRLAB? It's a relatively small drug discovery company. We're about 30 people, and we're focused on developing treatments for Parkinson's disease or different symptoms of Parkinson's disease. The company is an offshoot from the research group of Erik Werner, the discoverer of dopamine's function in the brain. And we have multiple drug candidates in preclinical and clinical development. Clinical development means testing in humans, and preclinical is everything you have to do before you're allowed to test in humans. And I'll talk about the clinical side and about how you can use Bayesian methods in clinical trial design, or one way to do it that we're working on. But just the basics are that usually you do a randomized control trial. We have one control group and one or more treatment groups. And then you define a primary endpoint, which is some kind of measurement that's supposed to characterize your if your treatment works. And usually you have some kind of classical significance test to see if it's comes out positive or not. And crucially for the rest of this talk, it's possible to update your what test you do after you have received some blinded data back. So you have to pre-register and pre-specify your analysis before you see unblinded data, so before you know which patient was in the treatment group or in the control group. And then usually you measure lots of things. You define pre-specified secondary endpoints, and you can do post-hoc analysis after the fact. But this primary endpoint is very important, and for many people, the takeaway from the study is just was it successful or not based on this, whether this primary endpoint comes out significant or not. So it's very important. If we have a drug that's working, then it's very important to try to not get a false negative on this, because it's very bad for the development. Of course, we also don't want false positives because we want to find the truth. But we'll come to that. Okay, so the rest of the talk will be a little bit about how one way that one could use a Bayesian model for doing the study planning and blinded data review. And this is work in progress. So we're developing these models, and we would be very grateful for feedback and discussions. All right, so from now on, I will talk about a simulated case study. So I've just made up hypothetical treatment for some kind of event. We assume we have a candidate drug, which we believe reduces the frequency of some bad event. And we think that we might be able to reach a reduction of 40% compared to placebo. And we want to detect such a reduction. This is clinically relevant, probably. So this is sort of the assumption that goes into it. And then we have a list of uncertainties. So for example, what's the baseline distribution of this event rate in the target population? So baseline here means when you enter the trial, but before you get treatment or control. Placebo. What's the placebo effect? And what's the level of individual variation? So we say that there's an average reduction of 40%, but of course it varies from patient to patient. And yeah, so we will have these uncertainties, and we want to sort of include them in the model to be able to make a good estimates for how the study would run. And there are lots of more complexities that if you were to do this in the real world for a real study, you would try to include as many of them as possible in your model. But here, to keep it simple, I have tried to remove all the complexities that I could. And again, yeah, this is just a simulated data from the rest of the study. Okay, so probably considering the crowd, the audience here, I should have written equations instead of text here. So I apologize for this, but hopefully you can also understand ordinary English. So what we've done, the way I specified this model is that we have measured some events at baseline, and then we measure. Maybe I can point with the mouse. Can you see?
Yeah.
So we measure some events at baseline and some events during the study. And these are drawn from a Poisson distribution. So what we really care about is some kind of underlying rate for each patient. during the baseline period and the study period. And then we assume that the baseline rate is drawn from some distribution with unknown mean and unknown standard deviation. So here the variables in red are the hyperparameters, the global parameters that define the model and that we care about. Basically, the parameters we care about. Okay, so that gives you the baseline rate, and then you assume that the study rate is given by taking the baseline rate and then multiplying by some kind of factor, the rate change. And this change is in turn drawn from another distribution, and it's drawn from one distribution if you're in the placebo group, another one if you're in the treatment group. And the placebo group has a mean change that we call, on average, the average change we call the placebo effect. It's a little bit confusing here because if it's a placebo effect, if this parameter has the value 0.9, it means a 10% reduction. So it's always this 1 minus 10. I think it'll be okay. And then we have the assumption that you have for the treatment group, you have this is on top of the placebo effect. And here we assume and fix the treatment effect to 0.6, so a 40% reduction compared to placebo. And then we assume that there's some kind of dispersion, so both the treatment group and the placebo group have some standard deviation, which is the same. So for this event rate. And sorry, not the event rate, for the change in the event rate. Right. And in code, it looks like this, where I've specified this model in PyMC, in Python. And I've highlighted the 5 lines that define these, the 5 hyperparameters that set up the model, basically. And here I've just assumed that they're all drawn from gamma distributions, except for the treatment effect, which we fixed. That's just to keep things simple. And here I've made up some numbers for the priors for the hyperparameters. This should be, in the real world, you would base these numbers on some kind of prior knowledge about what you expect. All right, so that's the model. Except for some detail, which is that I want to truncate this data generating model at 1 for the baseline rate. Sorry, the number of events at the baseline. If you get patients that don't have any events during the baseline, then it's hard to show a reduction. So we just exclude them from the study, and that's then included in the model here. In a way. Okay. So let's see how much time. It started 5 minutes late. Ish. I'll try to do that. Okay, so that's the model. This is the initial model we set up before we get any data back, which is supposed to capture all the uncertainty we have about our best guess for what might happen when we run the trial. And now we can sample the 1,000 draws from the prior predictive. And then based on this, we can then do a power analysis and see what are our odds, if our priors are correct, of finding a significant result. That's shown here on with 2 curves. On the left-hand side, we see the traditional comparison to 0.05, so 5% significance level. But I mean, it's being below 5% is better than being above 5%. But it's of course a lot better if you can get higher significance. So here on the right-hand side, I've compared with the stricter condition of 10 to the minus 3. So basically, if you're just below 0.05, this means that your uncertainty, your classical confidence interval only just excludes 0. So it means that you have very little understanding about the size of your effect. You just know that the sign is in the correct direction. So you ideally want the better significance than this. What else? I used in this case a negative binomial regression model to do the significance setting. This is quite standard for this kind of count data or event data. And here the dashed line at the 150 is supposed to indicate that it's been decided that we will run the trial with 150 patients in total. Ideally, of course, to get a good power and to be able to get good estimates, we would like to run a lot larger study than this. But it's often not possible to run as large a study as you would like because it's too costly and it takes, the more patients you have, the more time it will take to run the study. And also there's an ethical issue that you're not allowed to run a very large study for a drug that you haven't characterized well before, because it's an experimental. Right, so that was all before the imagined study starts. And now let's imagine that we get some blinded data back. Let's assume that we get it back after we have the first 50 patients have completed the study. And blinded here, of course, means that we don't know which patient was from the treatment group and from the control group or placebo group. So we only get these events for each patient. And in this case, I generated this table by simulating from a modified version of the model, where I fix all the parameters to some value, and I change some of the gammas to some other distributions, so log normal distributions, just to get some kind of mismatch between the data generation and the analysis. Okay, but how can we use the data to improve the model? Well, of course, this group assignment of which patient comes from which treatment arm, this is just another uncertainty that we could include in the Bayesian model. And in fact, it's basically just a one-line change. In this case, it's a three-line change, 4 if you include the comment. So this group assignment variable is assumed to be drawn from a Bernoulli distribution with the p of 0.5. In other words, for each patient, it's up or equally likely that the patient comes from the treatment arm it's in the treatment arm or in the placebo arm. But then I've added an extra soft constraint here, which is supposed to capture the fact that this constraint ensures that the number of patients assigned to the treatment arm never strays too far from 50%. And the reason for this term is that when you design these studies, you usually have stratification that makes sure that throughout the study, this constraint is satisfied, basically. And I want to, so if that's in the data and I know it, then I want to have it in the models. Also here, I've changed, instead of having a constant treatment effect of 0.6, I've now allowed it to change ever so slightly. I mean, it's a very strong prior anyway, but this is basically a way to monitor if the model wants to change the effect or not, to see if we have anything there. Okay, and then we sample from the model. And if you do this, you will see that the PyMC it samples fine, but it uses a little bit strange compound step. So it uses this efficient nuts sampler for all the continuous parameters. But this group assignment variable is discrete and it cannot use a nut sampler, so it has to use a less efficient metropolis sampler. But it's for such a small data set and small model, it runs fast enough anyway, so it's not really a problem. But it means that if I understand correctly, it means you wouldn't be able to run this model in STAN. Then you have to modularize away these parameters, and that would be very complicated to do when you have this constraint that I added to the model. But this allows me to show you a joke that I like very much. It's in Swedish, so I apologize for those who don't understand it, and I also apologize to those who do understand it. Okay, anyway, so right now, that was the model, and now we can look at the posterior for this model. We have these 5 parameters that define the model, 2 parameters for the baseline, and 3 parameters for the what the effect what happens when you go from baseline to study period. And what you can see here is that, oh, and also I've added here the true value because I've simulated the data, so of course I know the true value. So you can see what we managed to learn. And what you can see is that for the baseline, which of course doesn't care about this, what group you're in, it's the same for both the treatment and placebo group. You see that you managed to constrain the values quite well. In fact, you also managed to learn a little bit about the placebo effect and the dispersion, the amount of difference between patients in how it changes when you go into the study. So parts of the prior have been excluded here. So we have learned something. Yeah, so there's a higher baseline rate. There's more spread in the baseline than expected, and there's also more spread between patients and what happens when you go to the study. So if we now use the posterior instead for our simulations, we can see what happened to the chance that the trial will come out positive. And what we see is, unfortunately, we have lost a little bit of power. So the dashed line here is what we had before, and the solid line is now what we got after we updated the parameters. And the reason is that this negative binomial model is relatively sensitive to misspecification, and especially when we added when the dispersion in this study change rate, when it grew, this is means trouble for the negative binomial model. So instead we try another model. It's a very simple one. You just look at this number of events during the study over the number of events during baseline, which is basically a noisy estimate of what we care about, namely this rate change. And then we rank that and run ordinary least squares. And if you do that instead, you see that instead of the decreasing power, the power shoots up now. So we have a larger probability of finding a positive result according to this study. And again, in this case, I've simulated the data such that there is an effect there. So that's why it should go to 1 when you go to have an infinite number of patients. What else? Yes, and the reason why it goes up here is because the number of events at baseline was larger than expected. So this means that this measure is less noisy than we thought when we started the study. So you have a better precision in your estimate. And this model is not as sensitive to the noisy intergroup variability as the negative binomial model. All right. Finally, then, let's assume that the trial is over. And now we get data for all the 150 patients. And the question is now what happens? And here it's again the same as before. I showed you the blue and the orange curves, which was the prior, and then the analysis after 50 patients had come in. And now we have 150 patients, and I want to show the difference between the blinded analysis with 150 patients, which is the green one, and then the full unblinded analysis, which is the red one. And basically what you can see is that it turns out for this particular case, we managed to learn a lot about the baseline parameters and in fact about the group dispersion without unblinding. And we learned a little bit about the placebo effect without unblinding, but for the treatment effect, the interval didn't shift at all before we did the unblinding. And then we saw some shift and it shrank a little bit, but not so much. I mean, in this case, the prior was very tight already, so that's the reason. And 150 patients wasn't enough to narrow it down further very much. Okay, finally, some sanity checks. We saw that we expected a better power with the ranked model, but if you simulate from the data generation procedure, do you really get significance more often? And it turns out that indeed you do. It's a little bit busy here, but the dashed curves here is what happens if you sample 1,000 trials from the true data generation procedure. And in that case, in fact, both of our model was a little bit pessimistic, and the true model gave significance more often both for the negative binomial model and the ranked model, but the ranked model is less. And finally, what happens if the true effect is 0? So in this case, if the treatment effect is at 1, which means no difference between treatment and placebo. So in this case, it's a bit interesting. What happens then is if we have this very strong expectation that the treatment effect will be 0.6, then this doesn't shift very much at all until you unblind the data and inform the model that actually some of the patients were from the treatment arm. So instead, what happens is that the value for the placebo effect shoots up. And in fact, if you look at the green one, which is when you have 150 patients but still blinded, it thinks that the placebo patients do worse instead of slightly better, which is the truth. And the group dispersion here is a lot smaller than it should be. So the true value is over here, but it's estimated as being a lot smaller. So basically what happens is that what we really have are two distributions that are on top of each other, the placebo and the treatment one. But because I have this strong prior of a separation between them, this gets interpreted as two distributions with a narrow spread. So therefore the model is updated and it's very enthusiastic now and thinks that, we have a lot of baseline events and we don't have so much dispersion between groups, within the groups, sorry. So that means that we have very high possibility of getting significance. But of course, if you test this with the real data generation procedure, where you have a null effect, then in fact you do get, if you look at the ranked linear model, you do get false positives only 2.5% of the time, as you should. I forgot to mention what the star here means. It means that actually we take for two things. We should have a 5% p-value below 5%, and it should be in the correct direction. So this should happen around 2.5% of the time. And in fact, actually, with the negative binomial model, you overestimate the number of significance, or you get significance more often than 2.5% of the time. And that's because of this mismatch between the models. So if you have these extra outliers, then this, by the negative binomial model, it assumes that these come from a narrow distribution, and then it thinks that there is a signal even when there is none. So in this case, the ranked linear model is certainly better because it both has a lower false positive rate and a lower false negative. Right. So that's the end of the talk. Yeah, this is a work in progress, and I'd be very grateful for feedback. And if anyone wants to discuss this or anything more in general about how to use Bayesian methods for clinical data planning and analysis, it would be very interesting.